Are you ready to embark on a journey of success? To discover your true potential in the untold stories of careers in life sciences? Welcome to Pathfinder by the Purdue Institute of Inflammation, Immunology and Infectious Disease. With your host, Tommy Soares, discover the untold stories of industry scientists and unveil the secrets to excelling in your career. Thank you for joining us once again, and remember to hit the like button and subscribe to our channel. Welcome, everybody. My name is Tommy Soares. I'm the Director of Scientific Strategy and Relations for the Purdue Institute of Inflammation, Immunology and Infectious Disease. And today on Pathfinder, I have the honor of introducing you to Dr. Muriel Eaton, a PhD graduate from Purdue's Department of Medicinal Chemistry and Molecular Pharmacology, now serving as a clinical scientist at Med Institute here in beautiful West Lafayette, Indiana. Dr. Eaton came to Purdue with a Bachelor's of Science in Biochemistry from Kansas State University. During her time at Purdue, Dr. Eaton characterized a new mouse model of SCN2A genetic neurodevelopmental disorders and leveraged that model to further understand the molecular mechanisms and directions for treatment. Her dedication to both her studies and education earned her the Jenkins Knevel Award for outstanding graduate research from the College of Pharmacy. Welcome, welcome, Muriel. Thank you so much for being part of our Pathfinder program and helping us lead the way today.
Hello, it's good to be on here.
It is wonderful to have you. You know, I've been trying to follow your career trajectory. I know we have your interview captured from when you were at Purdue as a graduate student for our Purdue Lecture Hall series. And, you know, I've been so curious to know about that transition, how you got the job, how was the whole interview process, what are you doing now as a clinical scientist, and what are your responsibilities? What did you not know then that you wish you knew now? All of those good things. So I don't know, as far back as you want to take us, you please, the microphone is yours, and I will poke and probe and ask and try to unpack more as we go.
So I think the best place to start is really how I got my passion and my love. So we did a cute little science experiment in first grade. We put a penny in vinegar, and it was shiny. And that was just so completely magical to me. And I was like, I love this science thing. I need to keep doing this. I need to learn about this. And we did the Science Olympiad, extracurricular activities, Bio Club, and unfortunately, and when I was 10, I started getting really debilitating migraines. And I'd skip most weeks of school at a time, maybe going for a little bit and have to get taken home again. And so that kind of really drove me. I'm just like, you know, there's got to be something to help this. And I really want to learn more, like what's really going on. That also kind of taught me to take advantage of, you know, the time that I was feeling good and working hard and learning more about science in general. And so for my Gold Award Girl Scout project, the equivalent to the Eagle Scouts, I created a program for inner city schools to have more of those hands-on science classes. So I could spread that passion and people could learn more about the magic of science. And even through college, I did chemistry magic shows for local community groups, just explaining more of that magic of chemistry. And so I went to Kansas State for a bachelor's in biochemistry. And I guess I should have started before that, because I was a junior in high school. I went to a lecture. The Fungal Genetic Stock Center was in Kansas City. And so they're a main repository of various fungal strains, which is really important so we can study different things and different organisms and different strains. And so afterwards, because during the lectures, he said he was looking for students, and I was a student. And so as a high school junior, I went up and said, I want to learn about PCR. He had some funding, and I actually ended up getting a paper published in high school through that. And then crazy thing happened my senior year of high school too. There was somebody's brother-in-law was the director of vaccine research. You may know him as Dr. Barney Graham, who was Time Person of the Year for his work with COVID. And I actually did some bioinformatics projects for him and his team to study respiratory syncytial virus, which currently has developed into a vaccine now.
Awesome.
So get your vaccinations for RSV. So I was kind of just starting up in college, and the summer before I started classes there, I found a lab studying proteomics. And so we looked at protein degradation of the proteasome in the yeast model. And I really like that. I love working with the cells. I love doing the lab work and analyzing the data, designing the experiments. And so I did that all throughout my career. And then I also learned some more genomic information. I joined a lab studying CRISPR in my later years in undergrad, finished undergrad in 3 years with all of my AP transfer credit, and started grad school 2 weeks after I finished my summer undergrad research program, and jumped right into it, looking more at neuroscience and really learning about neurodevelopmental disorders, and studying kind of the rare diseases has really turned into a big passion of mine, because they're mostly underlooked by the community, but have detrimental effects on quality of life for some of these patients, kids mostly. And I knew I wanted to do science. That was a given, but I didn't know what to do with my science.
Yes.
And so I thought about different things. I thought about forensic science. So I joined the Explore Post program, learned more about the police force to try to be like a forensic site investigator. Then I was like, I don't really want to see bodies and crime scenes. So I was like, that's not for me. Then I tried doing the business route. I really like strategic consulting, kind of getting the plan together of doing a study, doing a project. So with Purdue, I did the Applied Management Principles Certificate program. And there's a lot of things in business that are not science. And I was like, stay away from politics. Okay, I'll try something else. And I always wanted to teach, and I was a TA in undergrad. And so I did the foundations in graduate teaching. That's offered at Purdue. I got that certificate as well, and was thinking about the teaching route. And then COVID hit, right? Right before I was graduating. It was great for my dissertation writing. Don't get me wrong. I could stay home. I fostered 9 puppies, and it was a blast.
Wow.
It was very hard to find a career, and I still didn't know what I can do like with science. I'm just like, I like science. What do you like about science? I like writing. I like analyzing the data. I like planning. What can I do with that? And so I reached out to my LinkedIn network, some other people at Purdue, and found Cook. So Cook is one of the major medical device companies, came up with the first heart stent. A lot of cardiovascular innovations come from Cook. And my friend was a medical writer there. And I said, that sounds great. Kind of want to deal with patients though. And so I found the perfect position for me, and that is a clinical scientist. And so what I do is I help in the planning phases of clinical trials and clinical studies, getting all the FDA required documentation, the protocol, the kind of what the case report form data collection forms look like, getting the data management system set up. And then the medical doctors do their thing. I'm a paperwork doctor is what I tell people. That's my doctorate in paperwork. And then I get the data back, quick analyzing it, checking it, making sure it makes sense and kind of greater scheme of things. And then getting it all wrapped up in a package for FDA submissions.
Nice.
And that's just kind of my trajectory in a nutshell. And then I've also the scientific communications team. So I help write publications, clinical evaluation reports. So in the European Union, you have to basically defend why your medical device should still be on the market and just, hey, it's still safe and effective. We're doing okay.
Mhm.
And so writing those documents, doing lit searches. So I'm still doing my writing. I'm still doing my data. I'm still doing my science, and still helping in the big picture, still helping people. So
That's wonderful. Wonderful. It sounds very fulfilling. It sounds very much like a great fit for what you seem to have gravitated towards, which yeah, I know you had tutoring and this type of grant writing and helping with the writing piece. So it sounds like it's a wonderful fit. I want to go take you back a little bit so that you can give us also some juicy bits here of what transpired. So, okay, you said you saw vinegar glow when you mixed it with pea. Is that right?
it was a penny. It cleans the
Oh, with a
penny.
penny, a
Mm-hmm.
penny. Okay, with a
Yep.
copper. Oh, awesome. Okay, okay, cool. Did your parents ever buy you like a chemistry set or anything like that when you were a kid that you played with? Or was this just it, the first time that you got to see it?
That was the very first time, but my parents always set me up. Um, just kind of, just what you're interested in, we'll support that. And so my dad was the coach of the Science Olympiad team.
Oh, awesome.
And so that was a really great opportunity. And they have that program here in Indiana, a lot of different events and scientific fields. And there's like a building component. So I was in Science Crime Busters, which you do have like a pretend crime scene, and you do paper chromatography and do powder identification. So that was really fun, like science competition
Yes.
that my parents supported. And I did get one of the microscopes that plugs into your computer with the USB. Get a
Awesome.
look at things. Really like molecular biology. And yeah, my family was very supportive, and the school system in Kansas too is
Yeah.
very STEM focused as well. And so that's why they had the opportunities. But sometimes you just have to go seek them out. And you're still in high school and you go to these lectures, you never know what's going to happen. The two lectures I went to, I got internships out of,
That
which
is
is crazy.
awesome.
So just being involved with the community and being willing to kind of put yourself out there to learn.
Yeah, I think that's such a great lesson for everybody to hear and to listen that, unless you get out there and start meeting and becoming part of the community that you want to be a part of or that you're interested in learning more about, you got to get out there. You have to ask questions. You got to stick your neck out a little bit and take a bit of a chance. Nothing bad can happen. Nothing bad will happen if you do, and only potential great things like two internships when you do these things and you get involved and you start, becoming interested and curious about what can I do? What can I be involved with? What an awesome opportunity though to be involved in research that produced a publication in high school. Like I don't know that I had the maturity then to understand what this thing was all about. You got a real like a real lesson by doing and by being part of that. Tell us a little bit, what was the environment in that lab, and were there grad students? Were there other students that you were working with, or was it mainly with the prof?
So the Fungal Genetics Stock Center is owned by a lot of grants, and so they maintain and get different strains of fungus, Raspberry crassa, common one. And so our project, so he got its grants, and so part of it was connecting with the community. And so he brought in a couple of other students and me, and so I worked with the curator of the center and one of the other major assistants there. And we helped characterize one of the tryptophan synthase genes of some different strains and looking at the sensitivities to it. It's PCR and gels and looking at more of genetic understandings of the different loci in that fungus. And that's where, I really got my start learning how to do PCR, learning how to run gels, um, use a pipette.
Yes.
It
Yes.
was whenever we were walking around the lab, and I had my dad with me too. That's like my first time, like, oh, this is in fancy lab, Kansas City, and my dad was walking around with me. And he goes, wow, that's really cool. What's that? The curator goes, that's a test tube holder. And so just like learning a centrifuge, how to balance a centrifuge, just very
Yeah.
fundamental stuff with
Yeah.
more advanced lab equipment that yeah, I know how to use a beaker, a graduated cylinder, kind of the basic equipment,
Yes.
but some of the stuff you don't normally see until, you're in a lab. And so I got
Yeah.
to have early exposure to that kind of thing. But even like I had an independent science fair project that I looked at worms. So I called a professor, cold called, because my grandma just got diagnosed with Alzheimer's disease. And I was like, that's my science fair project. I'm going to study how the FDA approved drug Donepezil for Alzheimer's it affects with all these different supplements that she's taking. So the flavonoids, ginkgo, CoQ10, like everyone takes it, but nobody reports it to their pharmacist. Like, oh yeah, I'm on the supplement. Well, it actually interacts with the FDA drug and makes it not perform as well. At least that's what I saw in my worm models. But called a professor in Colorado that was working on it and said, hey, I see you have Alzheimer's worms. Tell me about that. What do I need to do? And my mom drove me to the local brewery shop, and I got agar plates and all the stuff to make agar plates to grow the worms.
No
Um,
way.
but
That's
you just have to you just can't be afraid of failing. I think that's the main lesson that I've learned in asking silly questions of, hey, what's that fancy piece of equipment?
yeah, totally, totally. No, and absolutely, you have to kind of come out of your shell and ask those questions. I am still in awe of what what a broad horizon that gave you, that experience gave you. I mean, to have again, like you're impressing me because to have the maturity to know that I'm going to call a professor because I found out on one of their publications that they had these worms and I wanted to do this type of testing. Like that you really don't get to that maturity level mainly in graduate school, right? It's when your prof says, "Oh, why don't you call them up and see what they did on that paper?" And I think it's great and wonderful for everybody to hear this type of experience that you had because I could tell that it really opened up doors, but it opened up also your ability to really become a good scientist, a resourceful person that can go beyond the boundaries of their confines and say, oh, okay, there are people out here, they've done work and I want to build on this. And wonderful, wonderful. I really I want to ask you about the Barney Graham experience too, and what that was all about. Uh, because there's such great work that has come out of his lab, and I want to know about the experience there and the environment that you experienced during that time.
So that was from a lecture. It's just, and again, he was talking about careers and he talked about NIH and how he's like, yeah, we're kind of academia, but more fast paced, and kind of explain what the NIH was. And I didn't really know the guy, but he was a business teacher's brother-in-law. He gave a lecture and he talked about how they use computer programs to simulate and model different molecules. You can look at interaction sites for antibodies to develop vaccines. And afterwards, I came up to him with my little notebook, and I like got my questions written down and marched right up there. And I go, Doctor, I have some questions for you. And I forget what the question was. I was like, oh, doesn't the stabilizer you use for imaging going to affect the conformation of the protein structure and hide pockets for, targets for antibodies? Something along those lines. He was like, oh yeah, that's a great question. And, here's how we deal with it. He's like, how did you know how to ask that? I was like, well, played around in molecular modeling in one of our classes and the student version of JMOL, PyMOL, and all that. And he goes, I'm going to need some help. And I was like, okay. And he's like, how do you feel about learning another program? I was like, sure. And so I got a remote unpaid internship for a year. I worked with some other members of his team. And I mean, I had phone calls with him too. And also one of, his counterparts in Spain that was also working on a similar, RC design. And so he had structural conformations of the pre and the post fusion glycoprotein. So that's the protein that virus needs to fuse with the cell and to get in and spit out the DNA. So if you stop that, you stop the virus from getting in the cell. And that's really what you're looking for. But and it's really hard to capture and find a target once it's in that conformation. But during that conformation change, it opens up a lot of pockets. And so being able to model that change and say, okay, I see, where we could fit that, what approximate size we're looking for, what properties that we need. So you can take that mass screening library, narrow it down a bit, at least, pick something, pick a couple libraries to run screening assays on. So it's very basic work. Even just being able to model the conformation change, just predicting it,
yes,
can help inform and look at the kinetics of it. So it's very basic educational things. I'm not doing groundbreaking work, but I was still able to work with the team. And one of the graduate students from Urbana-Champaign, walked me through and taught me visual molecular dynamics and how to simulate these changes. And then once we had the final confirmation movie of the changing, all of it, the rendering took hours. Dr. Barney Graham called me and he said, how would you like to come to the NIH and present your findings?
What?
Yeah.
How would I?
And so my first question was, yes, but can my mom come? And that was my first time in DC. I went to Bethesda. We got the whole tour. Awesome. Got to see the lab and everything. And then I presented the movie to him and the other expert in Spain. And that was just so cool. They have at the Smithsonian, they have the first genetic sequencer. And so I got to see that during my trip. Awesome. It was just very, very memorable and just concreted that idea of, getting out of your shell, getting out of your comfort zone, asking questions and just everyone wants to do well and do science and help people. Like we're all on the same page. We're all on the same team. So just figure out where you fit into all of that.
We better send him the recording of this interview. I don't know if you continue to remain in contact with him, but I hope the people that are thinking about going into his lab can hear some of the wonderful things you're saying about it. And what an awesome experience to have so early on in your career. What do you think that gave you in terms of like when you started to do grad work and when you started to do your thesis project? What do you think you were totally ready? Did you feel like you knew exactly what needed to be done? No.
It's a whole other world than a cute science fair project. And there's a lot of things, knowing what's out of your control. And sometimes science just doesn't work. I, ran PCR for 2 years straight, and then one day it just didn't work for 3 months. I did the exact same thing. I ordered the same stock. It just didn't work. Could never figured out why. And then just kept trying. You just keep trying. You try different things, and you're like, okay, maybe I should have thawed it out on ice, because usually I just stick it in my pocket. I'm like, let's not do that. Let's thaw on ice better, maybe. And I mean, those little subtle things can change your project. And so being able to fail and be okay with it and not take it personally, because that was my whole identity, and it still kind of is. I'm a scientist. I should know how to do a PCR. And when it doesn't go wrong, I'm just like, well, let's try to figure it out and move on.
Yeah.
So I think that's kind of the lesson I didn't get until grad school, because I kind of had that advocating for myself, PI calls it looking for trouble. Like asking those questions and be like, hey, has everyone had a project about this? Or we should probably look at motor deficiencies. So I was like, this is a neuroscience lab. I was like, yeah, but motor. Let's look at the gut microbe because there's a connection between the nerves in your gut and the brain. So let's look at that and just that I already kind of had built upon with my previous experiences, but the trying when you fail, the long, I wasn't ready for a full-time science job of some days you're just watching a mouse run in a maze, and some days you're counting poop. Like that's just how it goes. And you kind of have to step back and put it in the big picture. And with a lot of the rare disease communities, there's a lot of online forums, and you get to see parents, caregivers, their doctors even, and they're reading the basic publications because they'll take any information they get. Even these mouse models, they're not going to, base their whole management, but it's going to get some consideration. So we had a paper about sleep and neurodevelopmental disorders related to the gene for the sodium channel dysfunction, and just looking about, hey, maybe the time of medication may affect its efficacy. And so clinicians can use that and inform and say, okay, that's not going to hurt. Maybe pushing it back an hour, move forward an hour. That's a minimum change that make it, even if the patient can sleep an extra hour, that's a big improvement for some of these patients.
Yeah, no, and it's wonderful. I wanna go back to something you said that I think it's very true for many of us. And you said something like, you identified, or that you like, this is my image. I'm a scientist. I should be able to do a PCR. Um, and how many times has it happened where you have this image of yourself to only realize, well, it's a limited image. It has some limitations here, but this whole idea of this is what it takes to be a successful scientist, a failure-free scientist, is almost a bad idea, right? Like it doesn't work out well because we have to rely on other scientists, other people, and other people's perspective. It's that diversity that makes science so rich, and it helps us move through the problems. So what would you say to somebody that's having this identity crisis, thinking, oh my gosh, I should be able to do this recombinant protein expression. This I can't do any of, like I'm just not a good scientist, is what a lot of people end up saying to themselves.
And it's very hard too, and you kind of feel the weight of it because you're like, this is my dissertation, and but also too of like, hey, these patients are waiting on this data, and you kind of feel the gravity. You're like, why can't I do things like maybe something really simple like that? And you're like, I should know how to do this, but being able to admit, okay, I may be doing something wrong. That's not going to change who I am as a person, even though that is a big part of my identity as I'm a scientist. And just kind of step back and like, okay, well, a good scientist, okay, I'm a scientist. What does a good scientist do? They ask questions. Okay, why isn't this working? They go find resources. Okay, I asked one of the undergrad, I said, follow this exact protocol. See if it works for you. Talk to other, use your network, use your resources, figure out why it's not working. And it called the company of the of the primers, and I said, okay, can you send me, the lot information from this? I want the certificate of conformity. Make sure it's, not contaminated. Make sure it's exactly what I want. Like, just try to, work the problem, be the scientist, and that's what a good scientist is. A good scientist is, I've built multiple things, broken many of the glass, where that's not what a scientist is. A scientist fails, and they ask questions why, and they try to figure out solutions and answer questions. And that kind of idea helps with the identity part of that.
And so I appreciate that so much. Is there a difference though in that identity, or in that idea of scientists and a successful scientist with certain, obviously, these challenges and hurdles that we need to superate. Is there a difference between an academic scientist and a scientist in industry, as you've seen kind of both sides now?
I think it's the same concept. We're all on the same team. We're trying to gather information. We're going to ask questions. We get hypotheses. The end goal is we want to increase global health. And it's just different ways of doing that, different perspectives. The industry is a little bit more translational. Things are a little bit faster paced, but you get to dig deeper in academia, really ask those mechanistic, fundamental questions.
Yeah.
We really need both, and we really need to use both. Eli Lilly has a lot of collaborations with Purdue. We did a collaboration in my lab looking at pain sensitivity of different buffers they were using in injectable, and how that affects pain levels of dorsal root ganglion neurons. And so, they have the resources, they know why, they know it's happening, but academia is really going to dig in and figure out, okay, why is it this? Oh, it's affecting the osmolarity, and that's activating the senses of the cell. Like, hey, I'm a different osmolarity. I'm going to start releasing things. And so, just understanding that pathway so they can make informed decisions about how to translate that. So, we really need that both sides. It's the same work, it's the same science, just done a little bit differently.
Yeah, I appreciate that answer a lot, because there's still very much same requirements, but the questions that you're asking are different in industry than you do in academia, as far as I understand. And having the ability from doing these interviews, I've learned a lot, having the ability to say, you know what, I don't know the answer to that question. What do you think? And how can we, right? In academia, if you don't know the answer, you're gonna be like, I'm gonna need to look that up for you and get back to you. Sometimes there is no time in industry, or there is no dollars that will allow you the freedom to go do a lit review for months on a
Mm-hmm.
particular topic, right? I
Yeah.
I I also am very curious to know, what was the transition like? Okay, COVID hit. Yes, you raised puppies. How many puppies, Muriel?
There's 9 puppies and their mom, and then another foster pit bull, and then
Oh my
the
God.
other dog.
Oh
There's a
my
lot of
God.
dogs. A lot of dogs.
Lots of dogs. But a
I'm
lot
a
of
cat
puppy
person.
chow. Yeah.
There's 4 cats at the house too at the time,
Oh
so it was,
boy.
it
Oh
was a whole
boy.
deal. But
Jeez. So, okay, so while you did that, and I'm sure you practiced your sourdough recipes like many people did, what was that transition to say, okay, I wanna be able to work in this company. I wanna be able to get my stuff together and start interviewing or applying. What was that process like? Can you tell us a little bit?
so it's kind of a different way to present your work because especially kind of this is more of a away from the bench position paperwork. My bench is my desk. Um,
Yes.
but and so being able to, because I have this knowledge, I can do experiments and being able to understand what we call the soft skills, and saying, okay, what have I, what did I do that can translate and I can be able to explain it, because it's going to be, you're doing the same work. You're designing a project, you're managing the project and your time, you're balancing that in resources. You got to understand, okay, I got this so much, especially with mice, you got to divide up the mice, and in industry too, you got to figure out, okay, this is what the resources I have to work with. So it's a very similar flow, but being able to express that in an interview and on your CV, that yes, I can do project management because I can do an experiment and you can manage that for 5 years. And then, on the writing side of things, you're writing publications and grants is kind of the, similar of some of the study reports that you have to do, for on the clinical side of things, and trying to translate, because you're going to be doing trying to find that connection. You're going to be doing the same work. It's just going to be phrased a little bit differently, just because you're not getting your hands dirty with pipettes.
Right, right. And so, okay, so there's a bit of an art form in relating that in your resume, right? But so did you just start to apply to many companies once you had that resume and it was nice and tight? Let's say you had all the information you wanted there expressed in the way that you wanted to. You're mentioning this position. This was your focus. This was the position that you really wanted to get, and so you put most of your energy on that one.
So I actually did 50 applications. It was COVID, and a lot of places had stuff posted but weren't even hiring. I only heard back from about half of the applications I sent.
Oh, wow.
And a couple of them were, I applied to anything. And initially at first I was like, well, I don't feel qualified for this position. I don't do human work. I did one market study for Microsoft, like, sure. But I haven't done any human work. I'm not qualified for this position, and kind of getting in that imposter syndrome. And I mean, with COVID happening and everything, and trying to apply to jobs, you're like, you just don't feel like, oh, maybe I can just get a bench work somewhere, or work at a behavior core. But kind of taking that step, and I think having the support of kind of your friends and family of saying, like, maybe what other things you could try. And so I was reaching out to some other students. I was like, okay, maybe I like scientific communications and do some more medical writing. I still want data analysis, so maybe a medical liaison, being able to explain science to patients. That's really fun. That's like even teaching AP biology positions. Like, I was just
That sounds familiar.
trying to find everything. I was looking up a lot of different things. So all the way from postdocs, teaching.
Sure.
But this job was, I think, number 47 or something out of 50. I stopped after 50 applications. I'm like, there's got to be something in there. But it was just hard because of COVID. And this position really caught my eye, because it kind of definitely favored it by the other positions too at Cook. But this kind of had the best balance for me as a person and my interests, so that I can analyze the data, design the projects, write about it. Were really kind of my three areas of strengths and my passions. And also, Cook is a great company, just because they're not publicly traded too. So it's more, you can have that more patient focus. A lot of some other bigger companies still have that patient focus too. But kind of at our level and our position, and one of our smaller, we have a smaller umbrella company. And so we're able to really get involved more about all the different projects. And I've appreciated that. And
no, go ahead.
I was saying that the interview process with Cook was really it really impressed me, and I kind of knew I fit there because the president of the company sat across from me, and it wasn't just president of the company. He goes, "Hi, I'm Neil. I'm an engineer," and he went to Purdue, and like, he's just very humble person. And so there's just the people here that I was going to be working with, and he went back and forth with some ethical dilemmas that they ran into as a company, working with patients and these medical devices, and especially a lot of the new medical device regulations didn't really come into effect or were implemented until the 70s. And so these historical hypothetical scenarios that the company actually went through was really important to me to be ethical too about kind of our work. And a lot of companies are, but having that be a central focus too is really important for me.
Wonderful. And so tell us a little bit about Med Institute and Cook, and how does that relate to the other? Because you've been mentioning a lot Cook, but I want our audience to understand the relationship between Med Institute and Cook.
So Cook is kind of the big parent company, and that's the cool pyramid building that you see here on the west side, Walmart. And so we have within that big Cook group, we have different companies to help focus on different areas of research. There's Cook Advanced Technologies, which mostly focuses on the engineering side of new products, and they kind of have a focus of innovation. And So there's some other companies in West Lafayette and through other cooks that kind of focus on a specific group, urology, mostly cardiovascular, and Med Institute, and that's kind of why we have a different name, is that we are more of a contract research organization. So because we have the specialty of cook and we have this wealth of knowledge, we don't want to limit it to just cook. So we take, we help our company cook. We also help other companies as well. That may be direct competitors, maybe because we just want to help people. There's different ways of doing that, and that's fine. We have all the firewalls and all the NDAs in place, so that way we can help more people. We can help the community because we're specialized in this. We know this. I say we. I just do the paperwork. Our company is, and so we bring the expertise of cook to other companies.
Yeah. Wonderful. That's amazing. And having this free agent type of freedom, right, to work with other companies, it must be also so fulfilling because you get to see so many different products, so many different types of trials and innovations that are happening. Muriel, what would you say you wish you knew then that you know now before getting going and starting in this position in industry? And what I mean by then is your time in academia. What do you wish you would have prepared for more to hit the road running, let's say? I'm not sure that you did or you didn't, but what would have been helpful?
I think, the culture is different. I wasn't used to sitting at my desk for 8 hours a day and walking around for meetings. So just kind of lifestyle. I got little pedals underneath my desk, and that I don't need to drink 3 Mountain Dews a day like I did in grad school. So it's kind of like basic lifestyle changes. But as far as like good lab practices, so there's a lot of regulations in clinical. And I didn't really understand or appreciate that in academia, because as long as you do good science, robust science, document things. But and when you get really down to it, there's a certain way you have to document things, certain things you have to write down. And I'm embarrassed to look at my lab notebook from grad school, what I know now of all these regulations. Like, oh, I should have wrote that down.
Yeah.
It's all in your head. And even like, 2 weeks after I defended and I was wrapping up, another grad student was like, hey, I'm looking at your data. I had a question about that one. I was like, yeah, I think I remember that, but I was like, oh, something I should have wrote down, shouldn't I?
Yeah.
So just the habit of, even just kind of familiarizing myself with the level of documentation that, that industry lab research is doing, and just kind of understanding that and appreciating that. In academia, it's like, okay, I should be writing down, even if I see something weird about the mouse, like, okay, he has an abdominal cyst, that could affect something, it may not. I'm just going to write it down. Yeah. Like, just err on the side, writing it down.
Yeah. That's awesome. It's such great advice. Dr. Muriel Eaton, thank you so much for being our Pathfinder today, clinical scientist at Med Institute. It's been a pleasure talking to you, catching up with you again. Congratulations on all of your success. You're counting almost 3 years at Med Institute, and I can't wait to see what comes next in your career trajectory. Is awesome catching up.
Thank you so much. Appreciate the opportunity.
Absolutely. And I hope you have a great rest of your day.
Thanks.
Awesome. Bye-bye.
Bye.
Thank you for joining us once again. And remember to hit the like button and subscribe to our channel.